Our bus driver is still confused about how to get all these people on the other side of the section. He thought of an idea: why not call the security? Security might come and force people to move from one section to the other section. This might be the only way to move people from one place to the other. Security might facilitate this process of maintaining sufficient space in the bus. This is an example of facilitated diffusion. Facilitated diffusion is when an external force, such as carrier proteins and security guards in this case, helps move the particles from one place to the other. Facilitated diffusion is also called carrier-mediated diffusion because a substance transported in this manner diffuses through the membrane with the help of a specific carrier protein. That is, the carrier facilitates the diffusion of the substance to the other side. Now, we have a stubborn molecule as an angry uncle who does not want to go to the other section. The security will be carrier proteins that will request the molecule to move from one place to another. So what is the difference between facilitated and non-facilitated diffusion? We can draw a graphical representation here. Draw a graph and keep the concentration of substances at the x-axis and the rate of diffusion on the y-axis. If we talk about non-facilitated diffusion, when the rate of diffusion increases, the concentration of substances also increases. This means the graph will go straight for simple or non-facilitated diffusion. Next comes facilitated diffusion. If we increase the concentration of substances, the rate of diffusion can increase up to a certain point and then it goes to a fixed constant. Why do you think that is? To understand this, let us take an example. A pore large enough to transport a specific molecule partway through. When a binding receptor is on the inside of the protein carrier, the molecule to be transported enters the pore and becomes bound. Then, in a fraction of a second, a conformational or chemical change occurs in the carrier protein, so that the pore now opens to the opposite side of the membrane. Because the binding force of the receptor is weak, the thermal motion of the attached molecule causes it to break away and be released on the opposite side of the membrane. Now this passage is so narrow that only some molecules can pass through it. And that is why the graph shown remained constant at a certain point. There are several types of security guards facilitating this diffusion. Among the many substances that cross cell membranes by facilitated diffusion are glucose and most of amino acids. In the case of glucose, at least 14 members of a family of membrane proteins, specifically called GLUT, that transport glucose molecules have been discovered in various tissues. Some of these GLUT proteins transport other monosaccharides that have structures similar to that of glucose, including galactose and fructose. One of these, glucose transporter 4, or GLUT4, is activated by insulin, which can increase the rate of facilitated diffusion of glucose as much as 10 to 20-fold in insulin-sensitive tissues. This is the principal mechanism whereby insulin controls glucose use in the body. Now, there is one difference in diffusion and the example of the bus driver that we used in this video. Getting to listen to the passengers can be hard, so the driver has thought of many different ways. And they may still not work. But diffusion is selective and will always work. We talked about diffusion and its types. We also talked about non-facilitated and facilitated diffusion. In this same series, we will be talking about one more way for the transport of substances through the cell membrane. Stay tuned and watch out!
Watch our medical videos anytime and anywhere. Download sqadia.com app now.